was an R&D assistant at JNCS Bangalore. He has a PhD from University of Manitoba, where he subsequently continued for the postdoctoral research, and later at University of Western Ontario, Canada, where he is currently working. Dr. Bhat is working in the field of cancer, metastasis, and therapeutic avenues. He has received numerous grants and awards for his research. The major awards are Simon and Sarah Gillis Thesis Award, Professor H.A. Nandan Gold Medal, Grass Advocate Choice Award, Gabriel Malapin Award for presentation. He has published his findings in reputed journals like Cellular Physiology and Biochemistry, Scientific Reports, Biocentric Cancer, Lung Cancer, Methods in Molecular Biology, and so on. Apart from his research, he is actively involved in teaching responsibilities at different universities in Canada. Currently, Dr. Bhat is a postdoctoral associate at the University of Western Ontario at Ontario, Canada. It is a privilege to have you with us, sir, today. Thanks for joining. Let me also thank Mr. Manjunath, who is steering the whole online session from UG College Studio. Mr. Ashok and Mr. Rajshekhar, who have been a great support for the smooth running of all sessions here. Dr. Bhat, we are ready to hear from you. Over to you, sir.
Okay. Thank you so much. Thank you for the kind introduction. I hear my own voice, so is there any way you could just turn off something? I'm not sure. Can you hear me now? Just give me a thumbs up.
Yes.
Okay, perfect. Sounds good. So thank you again. And before I forget, I would like to thank everyone. Thank you. I would like to thank the Institute, Mahajanas Education Society, organizing committee, and also the technical support. Especially, I would like to thank Ramesh sir for inviting me. It's an honor and pleasure to present my research there online, even though it's online. So without any further ado, I would like to start my presentation. Just give me a minute. I'd like to share it. Okay. I hope you can see my slides. Can you see my slides?
Yes, sir. Yes, sir.
Okay, perfect. Thank you. So this is the current situation. Sorry, is there any? Is it good?
Yes.
Okay. Okay, so this is the current situation. So it's day there, you have sunlight, and it's almost early morning, but still it's dark over here. And when you think about cancer, our understanding about cancer is somewhat similar to this situation. When you think about cancer, these are the things that comes to your mind: mutations, deletions, amplification, and epigenetics. And when you look at treatment options, all the treatment options are mainly based on information from these things. But there are certain things that are, I wouldn't say they are still in dark, but then slowly they are coming into light, and they are information about tumor microenvironment or metastatic microenvironment, immune microenvironment, which is very critical. And you have stemness, plasticity, and hypoxia. So understanding these things are very critical in addition to these factors. These are all cell intrinsic factors. So these are cell extrinsic factors. So understanding this helps us define or design better therapeutic strategies and also develop precision medicine. Now, what is precision medicine? It's a form of medicine that utilizes individual genetic information such as gene expression, protein expression, pathway alterations, etc., to prevent disease or diagnose a disease or treat a disease, and also in prognostication. Now, when you look at cancer treatment, the traditional medicine believe in one size fits all concept. So that means if you have a drug to treat one particular type of cancer, all the patients receive that particular drug. And what happens is that only a small percentage of patients, they respond to that drug. Some of them do not respond and others have adverse effect. So when you look at overall therapy response, it's not a successful, output or successful event or successful response. So in order to make it more successful, what you need is personalized medicine or a tailored drug. So this can be achieved by looking at their individual genetic information, gene mutation. And with that information, you can segregate patients into different groups, and you can design treatment for each of the group separately. And that way, what happens is that you have efficient treatment plan. Patients will respond better to treatment, and also you'll have lesser toxicity and lesser adverse effect. Now, when you think about precision medicine, it's a very fancy term and very enticing, but then it has its own challenges. And there are a few challenges. One is we don't have a lot of targets. There are only a few targets, druggable targets that are being validated clinically. And so there is a need for identification of newer targets or newer pathway alterations. Since I'm working on breast cancer, metastatic breast cancer, I'm mainly focusing on metastatic breast cancer here. Another challenging aspect is that identification of alteration that results in therapy resistance. Most of the patients, sometimes initially they respond better, but eventually they develop resistance to different therapies, such as chemoresistance or radio resistance. So we need to identify what results in development of resistance. And also, we don't have biomarkers to diagnose or for prognostication. Now, imagine when you go to a doctor with fever or something. So doctor uses stethoscope or a thermometer, or it does a physical examination to look at initial symptoms, and then based on your age, sex, weight, you will be given a drug. Eventually, you will be followed up. You will be asked to come back again. Again, you will be tested. He will use thermometer, and they will use stethoscope and other things just to look at the disease, whether it has become worse or whether there is an improvement based on the treatment plan. And if it is not working, then the treatment course will be changed. So there is a real-time assessment. I wouldn't say real-time, but at least you will have an option to assess disease progression or improvement. So, but unfortunately in cancer, you don't have a lot of biomarkers for prognostication or even for diagnosis for that matter. And the most important challenge is identification of mechanism that suppress immune system that's mainly induced by cancer cells. I'm sorry, here I wrote immune cells, it's actually cancer cells. That's immune system plays an important role, and I'll explain you why and how. Now, since my work is mainly focused on breast cancer, I'll talk about the different research that's been conducted in our lab and how the information from that research results in development of precision medicine. And hopefully at the end of the talk, I'll be able to convince you that some of the challenges that I mentioned here will be in a way not completely addressed. At least some part of these challenges will be addressed, and the results will be beneficial towards precision medicine. Now, breast cancer is one of the most commonly diagnosed malignancy throughout the world. You can see here the entire world is painted pink. That's because if you see pink, refers to breast cancer here. So that means it's a major concern. And breast cancer is a heterogeneous disease. So they display intratumoral and intertumoral heterogeneity. That means no tumor are alike. And also within the tumor, cells are not alike. There are different types of cells. And that poses a major challenge for treatment option and also could result in therapy resistance. So there have been developments, and molecular profiling has been conducted, and based on gene expression pattern, especially based on estrogen receptor, progesterone receptor, and HER2 receptor expression pattern, breast cancer is being classified into 4 different molecular subtypes. And these markers are now, they have become a clinical marker, and they use this in clinic to segregate or group breast cancer into different subtypes. Now, when a breast cancer patient comes in with breast cancer symptoms, what they do is that they perform a needle biopsy, which is a very painful process, complicated process. So they take a piece of tumor tissue, they prepare thin sections, stain them with these markers, clinical markers, ER, PR, HER2, and they also have a proliferation marker that is Ki67. And based on this information, they classify patients into different groups. So the luminal A, luminal B are hormone receptor positive tumors. Then you have HER2 positive tumors and triple negative tumors. They are called as triple negative because they do not, this type of tumors do not express estrogen receptor or progesterone receptor. So once the patient has been classified into one particular type of a group, the treatment is planned. So for ER positive tumors, they are treated with hormonal therapy. For example, tamoxifen, they compete with estrogen and prevent its binding to estrogen receptor. So estrogen it's critical in fueling cancer progression in these type of tumors. Now, patients who belong to HER2 subtype, they are treated with HER2 targeted therapy. You have Herceptin, trastuzumab, they are all FDA approved drugs. So they target HER2 receptor and prevent its signaling. Now, since triple negative do not express enough the receptor, these two treatment options do not work. So they are mainly treated with systemic chemotherapy. Sorry. With the whole intent of this is to manage the disease or extend their life rather than curing the disease. Now, there are other treatment options such as surgery and radiotherapy. They help in local control of the disease. And all this treatment mainly depends on size of the tumor and tumor burden. So if the tumor is smaller, then usually you go for surgery and followed by systemic therapy. If the tumor is bigger, then they go for systemic therapy, radiation, and then surgery. It depends on, again, size, stage of tumor, and also subtype of tumor. And some of the surgeons, in addition to removing the tumor, they also resect lymph node. That's the lymph node closest to the tumor, just to make sure the cancer has not spread to lymph node. When you look at the treatment response, luminal type, luminal A and luminal B, the estrogen receptor positive subtypes, even HER2 positive subtype for that matter, they have better prognosis as compared to triple negative. These are very aggressive and very metastatic. And that's one of the reasons why I'm working on triple negative breast cancer here. Now, this is one of the best examples for precision medicine. And I would say some people call breast cancer as poster child for precision medicine, because with these biomarkers or clinical markers, you can segregate patients into different groups and you can give them specific treatment option in order to get a better treatment response. Even with all this development and all this information, still when you look at breast cancer mortality, majority of the across the world, breast cancer is one of the most leading cause of mortality among females. And here in North America, you will see lung is the leading cause of death, but breast cancer is the second leading cause of death in females here as well. So what are the reasons for that? So one is resistance to therapy. As I mentioned earlier, some of the cells, they develop mechanisms to resist radiotherapy or chemotherapy. Then you have presence of residual disease. So after treatment, sometimes treatment do not get rid of 100% of the cells. There will be few cells, and they, in a way, form new tumors. So that could be due to either resistance or presence of stem with cells with stem-like characters. And these cells are very notorious. I'll explain you how and what can be done to control these cells. Finally, the major cause of mortality, more than 90% of breast cancer-related death is mainly due to metastasis. Now, what is metastasis? Metastasis is a multi-step process, is a very complex process where tumor cells from the primary site, that's breast in this case, escape and enter into circulation or spread to different parts of the body through either blood circulation or lymphatic system. When a tumor grows to a size of, say, 1 millimeter, the core of the tumor becomes really hypoxic. That's because they lack nutrient supply and oxygen supply. Just to overcome this problem, what tumors do is that they start secreting some of the factors that help in formation of new blood vessels. And that process is referred to as neoangiogenesis. In addition to that, they also release packets of information which contains these factors. They are referred to as extracellular vesicles. They play a key role in kind of remodeling the tumor microenvironment. They kind of influence surrounding cells in a way to facilitate further tumor growth and progression. And when the tumor grows to a certain size, some of the tumor cells that are around the periphery, they lose their cell adhesion molecule and start escaping from primary site, enter into circulation. And the cells that have entered into circulation are referred to as circulating tumor cells. Now, some of the cells die either due to sheer force of the blood flow, or sometimes some of the immune cells that are present in the blood, they attack them and then they kill them. So when that happens, they release their genetic content. They are referred to as circulating tumor DNA, and it has a clinical significance. I'll explain you in a while how these can be used in diagnosis and prognosis. Now, one thing I forgot to mention is that even though, when cells are in circulation, eventually they enter into a new site or distant organ, and then they survive there, form new tumors called as secondary tumors, and that is when the entire metastatic process is complete. So even though majority of the cells in the primary tumor are capable of escaping its primary site and entering into secondary organ, only a small percentage of cells survive and form new tumors. It's only 0.01% of cells survive and form new tumors. So that makes the entire metastatic process highly inefficient process. Now, the question is why? For that, we need to go back to 1889, where English surgeon Stephen Paget, he analyzed around 735 breast cancer patients. He performed autopsy on 735 breast cancer patients, and he found that the distribution of these tumor cells to different organs were not random. Instead, they followed a pattern. So it was an organ-specific distribution, and he explained that pattern through a hypothesis called as seed and soil hypothesis. He postulated that presence of right seed is referred to as cancer cells here, in a fertile soil, that is a favorable microenvironment, results in growth and colonization of cancer cells. Now, for a successful metastatic process, two factors are really important. One is presence of cells with stem-like features, or cancer stem cells. Now, cancer stem cells are known to survive even in a hostile microenvironment. And breast cancer stem cells are defined as cells with high enzymatic activity, that is aldehyde dehydrogenase activity, and also cells, and these cells express high CD44 expression. Now, another factor that plays a critical role in successful metastatic process is presence of favorable organ-specific microenvironment. So when these two things are present, then you could have colonization of a particular organ and formation of new secondary tumors. Now, when you look at triple-negative breast cancers, lung is one of the most common and deadliest site for breast cancer metastasis. So this suggested that lung might act or may act as a favorable microenvironment for breast cancer cells to survive and colonize. Now, there are a few studies that were conducted in our lab. Our lab was the first to show that breast cancer stem cells, they display higher propensity to enter lung and colonize. Later, we found that breast cancer cells interact with some of the soluble factors that are secreted in the lung, and that is essential for metastatic behavior. Later on, we found that lung microenvironment, they secrete some of the pro-metastatic factors, especially fibroblast growth factor 2, which is essential for lung colonization. So when we inhibited this, cells fail to colonize lung. And very recently, we found that breast cancer cells, they secrete extracellular vesicles. These are packets of information. They have protein, they have microRNA, they have RNA, they have DNA, everything. And these enter into lung and they remodel lung microenvironment and establish a pre-metastatic niche that in a way helps incoming breast cancer cells to survive and form new tumors there. So based on all this information, it was hypothesized that lung-derived soluble factors secreted, especially in the presence of primary breast tumor, they influence stemness, plasticity, and metastatic behavior. So to test this hypothesis, we generated lung condition media. We harvested lungs from mice that do not contain any tumor and mice that have triple negative breast tumors. Lung condition media was generated, and then that media was used to treat breast cancer cells, and different assay was performed. When we treated breast cancer cells with tumor-bearing lung condition media, we saw an increase in acquisition of stem-like phenotype. As I mentioned earlier, stem cells, breast cancer stem cells are defined as cells with high ALDH activity and CD44 expression. There was increase in expression of these markers through flow cytometry when we checked through flow cytometry. And we also found that the lung the factors in the lung condition media, what they do is that they inhibit expression of a tumor suppressor, that's DAC1, and through this it increases acquisition of stem-like phenotype. Now, when I looked at DAC1 expression in different subtypes of breast cancer, I found that its expression was significantly downregulated in triple-negative breast cancer as compared to other breast cancer subtypes. And also, when I looked at patient data, patients who have lower expression of DAC1, they have poor overall survival. So the black one, these are the patients with low DAC1 expression, and these are the patients with high DAC1 expression. So this suggests that they have poor overall survival as compared to patients with high DAC1 expression. So all these things suggested that DAC1 is a key factor that in a way suppresses stemness, and in presence of lung condition media, that suppression is inhibited. Now, DAC1 is also a transcription factor, which is known to suppress some of the key stemness-related genes, KLF4, CD44, and also epithelial to mesenchymal transition marker, that is vimentin. So in order to test if manipulating DAC1 did something to these genes, I knocked down DAC1 through siRNA targeting DAC1, and I found that deficiency of DAC1 resulted in increase in acquisition of, or increase in expression of stemness-related genes, and also EMT-related gene, vimentin. This was interesting because I saw similar increase when breast cancer cells were treated with tumor-bearing lung condition media. Again, this suggested a link between factors in lung condition media and DAC1 expression. This also suggested that stemness is key for metastatic colonization of breast cancer cells in the lung. So I wanted to test whether stemness is really important here. So to test that, initially I separated breast cancer stem cells from non-breast cancer stem cells through flow cytometry, and then we delivered into mouse lungs through tail vein injection. And lungs were harvested and cultured over a period of 21 days and imaged at different time points. What we saw was, one thing is that these cells express red fluorescent protein, and that's why all the images are red here. These are the cancer cells that are in the lungs. So as you can see, the breast cancer stem cells over a period of time progressed from single cell at day 0 to micrometastasis and macrometastasis by day 21. However, non-breast cancer stem cells, they did not show that pattern. This again suggested that stem-like feature is critical for metastatic colonization of lungs, metastatic colonization of breast cancer cells in the lung. So, because we don't have a lot of time, so I'll quickly summarize this part of the study. So far what we have seen is that the lung soluble factors, especially in the presence of primary tumor, they help in acquisition of stem-like phenotype in breast cancer cells. And they do it by suppressing DAC1 expression. And recently we have identified the factor that actually does this, that is fibroblast growth factor 2. And we have seen that inhibiting this prevents lung colonization. At this moment, currently we are looking at therapeutic potential of FGF2 and trying to see targeting FGF2 results in further suppression of lung colonization or prevention of metastasis altogether. Now, going back to my earlier slide, I showed you this data, not the data, these challenges, and in a way, I guess I can say that I have kind of contributed towards the first challenge, that is identification of genes or gene drivers or pathways that contribute towards metastatic breast cancer, metastatic progression of breast cancer. The next thing I want to talk about is identification of biomarkers, and that is why biomarkers are really important in diagnosis and prognosis. In this project, we are using different blood-based biomarkers to define a state called as oligometastatic state. So it's a phase 3 clinical trial, which is a multicentric and multi-histology. We are not focused on just breast cancer, but all types of cancer. Now, earlier notion was that metastatic cancer patients are non-treatable or there is no cure for them. However, in 1995, Hellman and Weisselbaum, physicians, clinicians, they identified breast cancer patients with fewer metastatic lesions, and they found that they responded better to radiotherapy. And they call these patients as oligometastatic patients. Now, very recently, we found that patients with just 1 to 3 metastatic lesions, and this study is called as SABRE Comet 3 because patients have just 1 to 3 metastatic lesions. They responded better to a specialized radiotherapy called as stereotactic ability radiotherapy. It's image-guided radiotherapy. And when these patients were exposed to SABRE, they had better overall survival and progression-free survival as compared to control arm, which was interesting. And this is the overall schematic of how stereotactic ability radiotherapy works. So first, a 3-dimensional image of tumor is acquired, and a high beam of radiation is targeted on the tumor, so that only a tumor cells gets affected, and it minimizes damage to the neighboring cells in a way, in order to reduce adverse effects in these patients. Now, the major challenge with oligometastatic state is that the state itself is not well defined. The definition of oligometastasis is not very clear. Some refer to it as patients with 1 to 3 metastatic lesions. Some refer to it as 1 to 10 metastatic lesions, and others say that 5 to 10 metastatic lesions are referred to as metastatic state. Now, for that, to define that, we need really good biomarkers. We can achieve that by taking biopsy samples, performing RNA sequencing or single-cell sequencing to identify novel biomarkers. Unfortunately, it's not feasible because, performing biopsy on metastatic areas is very complicated, painful, and also it's, in a way, it's difficult for the patients too. So the entire field is moving towards a non-invasive method, and that is referred to as liquid biopsy. Now, what is liquid biopsy? It's nothing but use of body fluids such as blood, saliva, urine, and also amniotic fluid to diagnose and prognose different diseases. Now, in cancer, blood, saliva, urine are used to identify and characterize different tumor-derived factors such as circulating tumor cells. I mentioned earlier that these are the cells that have escaped from primary tumor site and entered into circulation. Circulating tumor DNA, these are results, these are from tumor cells, dead tumor cells that are killed by immune cells. And then you have circulating immune cells. Again, these are the cells in the circulation, extracellular vesicles that are released from primary site into circulation, microRNA, and some of the metabolites, again, that are present in the blood or saliva or also in the urine. Now, since we saw a success in patients with 1 to 3 metastatic lesions who were treated with SABRE, now we have extended that study on patients with 4 to 10 metastatic lesions. Now, these patients are randomized to either to standard of care arm. So these patients get just chemotherapy, and another arm of patients, they get both a radiotherapy, that is specialized SABRE, and also standard of care. we collect blood samples at different time points, pre-treatment, during treatment, and post-treatment, just to assess whether treatment is working or not, or whether it is making it worse, or whether we need to remove patient from the study or not. So that will give us an idea, a real-time assessment, not real-time, in fact, but at least, I hope you understand, you could measure disease progression or improvement. So in this study, we are only looking at 3 biomarkers. One is circulating tumor cells, circulating tumor DNA, and circulating immune cells. So once I get the samples, what I do is that I take 7.5 ml of blood and use it for analyzing circulating tumor cells, CTC. So we have an FDA and Health Canada approved instrument called a cell search system, which is used to enumerate CTCs. So the blood is stained with different markers specific to tumor cells and immune cells, and we also use a nuclear marker. And this is how the picture looks. So the instrument takes images of all the cells, stained cells, and gives it to us. We sit and analyze each and every image looking for tumor cells. Now, these are the tumor cells. We call it tumor cells because they express tumor cell markers, cytokeratin, and DAPI. It's a nuclear stain. And if it's only cytokeratin and no DAPI, then they are not actual cell, that is artifact. And they should not express CD44 because CD44 is an immune cell marker. And this is how we identify presence or absence of tumor cells in the blood. And rest of the blood is used to collect circulating tumor DNA, ctDNA, for sequencing purposes. So what we do is that we collect those ctDNA and sequence them and look for pre-existing mutations and also mutations that are acquired during treatment. So that will give us an idea whether we need to change treatment approach or altogether stop that particular treatment. In addition, we also collect immune cells to look at activated immune cells. Now, one of the interesting things about radiotherapy is that it is known to activate immune system. So when radiation hits cells, they undergo lysis. So tumor antigens are released. Immune cells recognize those tumor antigen and start or elicit anti-tumor immunity in the area where radiotherapy has been introduced or in radiotherapy targeted area. In addition to that, these immune cells enter into circulation, go to different parts of the body looking for presence of tumor cells. And they recognize those and kill those as well, which are far away from the primary site. And this effect is referred to as abscopal effect, which is very fascinating and amazing how radiotherapy in one area could activate immune cells, which will eventually help in systemic eradication of tumor cells. So these 3 markers will be used in this study, are currently being used in this study to first define oligometastatic state and also correlate the differences with patient overall survival and progression-free survival. Since it's a clinical trial, I'm not allowed to share the data until the trial is done. Only thing I can say is that we have recruited 150 patients so far, and our target is 204. We are almost close to that, and we will be done with the Accural by mid next year, hopefully, and we'll have some interesting data. Now, at the end, before I wrap this part of my talk, I want all of you to think about this.
Sorry for the disturbance. I suppose the slides are not moving. Probably it's going on your system. It's not displaying.
Okay. Let me see.
Yeah, now it's visible. Yeah.
Can you see?
Yes.
Perfect.
Go to slides.
Yeah.
Yeah. That's okay.
Okay. So where was I? I just want you to think about this. Imagine a day wherein you just need a half a tablespoon of blood to identify oligometastatic patient and treat them with SABR to eventually treat and also potentially cure them. And this would be a really significant development in the field of precision medicine. Now, the final thing is the most important aspect, that's to identify a mechanism that suppresses immune cells. That's again induced by tumor cells. Again, I really apologize for this mistake. Please ignore that. Now, I want you to appreciate one thing. Cancer initiation is not a rare event. It's normal. And I'm sitting here talking to you. You're sitting there listening to me. There are cells in my body that are regenerating, especially blood cells, skin cells, colon cells. And when they divide, you have new DNA made. So 50 nucleotides are being added every second. So it's an error-prone process. Mutation could occur any time, and it happens every time. And it could occur in some of the critical genes, which could lead to cancer initiation. But it doesn't happen. Why? You walk outside, you have a biggest carcinogen above your head. That's the sun, which, the UV radiation could damage your skin cells, damage your damage the DNA, but still you don't get cancer. I know cells have their own inbuilt mechanism of repairing and also undergoing apoptosis if they fail to repair. But then what if those two things doesn't work? And that's when immune cells come into picture. They try to recognize those cells that have failed the first two process and they eradicate them. The funny story is that way back, cancer biologists did not believe that immune cells had any role to play in eradicating cancer cells in the body. And even immunologists, for that matter, did not believe that cancer cells attacks self cell. So tumor cells are not foreign cells, right? They are our own cells which went rogue. So even they did not believe. They thought, immune system actually targets virus, bacteria, other pathogens, and also cancer cells that are initiated by virus or viral they're called viral oncogenes, but they do not target any other cancer cells. But then later on, they started understanding when they found that some of the patients, especially HIV patients, where their immune system was suppressed, they started developing, majority of them developed cancer. And also patients who underwent transplantation, organ transplantation, again, they had a higher frequency, or they were at a higher risk of developing cancer because, again, their immune system was suppressed. Sorry, I guess I'm getting a call from message. Hello? Oh, is it? Where am I now? Okay, so you're able to see this? What about this? No, I just started with this, so yeah. Okay, yeah. Thank you. Sorry about that. Sorry for the technical issue here. So yeah, where was I? So later on, when they realized, some of the physicians, scientists, they came up with this immune surveillance concept. They said that immune cells, they look for tumor cells, they recognize neoantigens and then attack and prevent cancer initiation. But however, that was not sufficient to, in a way, explain anti-tumor immunity. But just 20 years ago, Gavin Dunn and Robert Schreiber, they came up with this cancer immunoediting, which kind of helped us better understand how immune cells recognize cancer cells and also how cancer cells evade immune system. So based on their hypothesis, they proposed that once cancer is initiated, tumor cells are recognized by immune cells. They start killing those cells, and that phase is referred to as elimination phase. And since tumors are heterogeneous in nature, there are different cells in a tumor. Some of the cells, they're selected due to selective immune pressure, and they in a way become dormant. They sit quiet without doing anything, without proliferating or anything, and that phase is called as equilibrium, the tumor dormancy or equilibrium phase. And eventually, after some time, due to some odd reason, they come up with a strategy to evade immune system or immune recognition, and that's when they start growing. And that's when you have cancer growth and progression. Now, I'm telling you this because it's really important to understand how tumor cells escape or evade immune system. And we also need to understand how immune cells actually recognize tumor cells and attack and kill tumor cells. There have been several studies, and this is one of the best example. That's identification of checkpoint inhibitors, discovery of checkpoint inhibitors by two Nobel laureates, James P. Allison and Honio. They found that the presence of inhibiting checkpoint proteins actually elicits anti-tumor immunity. Now, what are checkpoint proteins? These are proteins that play an important role in preventing overactivation of immune system. Now, imagine there is a viral infection or bacterial infection. So immune system gets activated, they clear clear the pathogens from our body. Now, after that, the immune system needs to be shut down. Otherwise, it will damage our normal cells too. So immune checkpoint proteins, what they do is that they prevent further activation of immune cells, especially T cells, and they undergo cell death. And that's how they kind of shut down the entire immune response. However, cancer cells, they hijack the system and make use of this system to evade immune recognition. And these two scientists came up with this inhibitor. They used antibody against checkpoint proteins, and they found that when they use it, the patients, especially this was tested in metastatic melanoma patients, and they had a better response. And these were some of the FDA-approved drug against checkpoint proteins, and most of the patients responded better to this particular checkpoint inhibitor treatment. This is something like the brake was released, the brake to suppress immune system was released, and due to that, immune cells started recognizing tumor cells and eventually eradicated them. This is one of the best examples for precision medicine. I don't know, this was published 6 months ago. I think many of you must have heard, it was all over the news. This clinical trial had around 14 patients. They mentioned 18, that's wrong here, 14 patients. And especially they had a mutation called as MMDR mutation. And when they were treated with this particular checkpoint inhibitor, they responded better to treatment. And all 14 patients, when they treated with this particular checkpoint inhibitor, they showed better therapy response, and the entire tumor just vanished, poof. So this was amazing. This was very interesting. But then it also told us that this works only on patients with MMRD mutation. Other patients do not respond to it. So there is a need for identification of more biomarkers, as I mentioned earlier, and more checkpoint inhibitor proteins or mechanisms that suppress or activate immune system or suppress immune system, so that we can come up with different strategies to either activate or activate immune system to kill tumor cells. Now we are taking similar approach in identifying mechanisms in tumor cells that evade immune response, and also we are looking at differential expression in tumor cells, differential expression in immune cells that is mainly due to presence of tumor cells nearby. And we are using a technology called as spatial transcriptomic technology. You could use different technologies. It's one of the omic technologies similar to microarray and single cell sequencing. But then if you perform this, you won't get all the information. For example, if you perform microarray analysis or RNA-seq analysis on the conference hall where you're sitting, it will only tell me that students are more in number as compared to others. When you perform single cell sequencing, it will tell me, okay, there are these many undergrad students, there are these many postgrad students, there are these many faculty members, these are the chief guests, and these are the technical support. Now, when I perform spatial transcriptomics, it will tell me who is sitting where. And it will also give me details. Say if a student is sitting next to Ramesh sir, it will tell me why that particular student is sitting quiet. And it will also tell me why Ramesh sir is actually keeping an eye on him. So I get all this information, and which is very beneficial in understanding how interaction between tumor cells and immune cells, and how tumor cells suppress immune cells or activate immune system. And the spatial transcriptomics work, this is an outline of how it works. We take tissue sections, as formalin fixed paraffin embedded tumor tissue sections. What we do is that we stain them with different markers. Here we have used cytokeratin, that's a tumor cell marker, CD45, immune cell marker, and DAPI, that's a nuclear stain, to look at identify cancer cells and immune cells. Now, then we select region of interest. We have selected 2 regions of interest, I'll tell you in a while. And these are hybridized with complementary strand, which has photocleavable oligos, and they are barcoded. And once that is done, UV is exposed, and they will cleave those oligos, and eventually they are collected and sequenced. So each oligos with barcode represent one particular gene. So if that barcode is red, then that means that particular gene is present in that. So that's how we'll identify gene expression pattern in this. Now, for this study, so far we have taken 19 triple negative breast cancer patient samples. They're all treatment naive. That means they are not treated. So that will give us an idea how how immune cells behave once treatment is started. So we have taken tumor areas with high tumor infiltrating lymphocytes, tumor areas with low TILs. So I'll call these tumor infiltrating lymphocyte TILs. It's too difficult to explain. And then we have matched lymph node sections. We want to see whether some of these tumor cells metastasize to lymph node, and also what gene expression pattern results in lymph node metastasis. And this is a picture of one of the section. As you can see here, the pink cells are immune cells. These are high in this area, and these are immune cells. And these are again immune cells that are low in frequency or abundance, and these are immune cells. So these are low TIL areas, these are high TIL areas. Now we are planning to look at gene expression pattern in both the cells that are close to immune cells, that are close to each other or away from each other, and also correlate this gene expression pattern with lymph node metastasis. Initially, we looked at distribution of immune cells, and what we found was that different patients have different immune cell distribution. I'm not surprised, because I mentioned earlier that breast cancer is a heterogeneous disease. Now, imagine one thing. Imagine there is only one drug to treat all the patients. What would be the response? Some of the patients might respond, some might not. So that's why, again, these things are telling us that we need to have a method to segregate these patients in a different group and tailor and introduce a tailor-made treatment plans, so that everybody gets are benefited, and everyone can be cured of cancer eventually. Now, I took some of the cells of interest and I looked at them, and you can see cytotoxic T cells are high in high TIL area, low in low TIL area. But interestingly, fibroblasts are high in low TIL areas and low in high TIL areas. Macrophages again show similar pattern, whereas NK cells are high in high TIL area and low in low TIL area. Now, this we are further analyzing these 2 cells, just trying to see if fibroblasts are activated or not. Usually, what happens is that in a tumor microenvironment or metastatic microenvironment, fibroblasts play an important role in fueling tumor progression, and especially activated fibroblasts. So we want to look at profiles of these fibroblasts to see whether they're activated or not, and also macrophages, whether these macrophages are tumor-associated macrophages or not. So this will give us an idea how to deal with different treatment plans and how it benefits patients. And then we looked at gene expression pattern in tumor cells that are in high TIL area and low TIL area. So the green dots here represent genes that are upregulated in high TIL tumor cells that are in high TIL area, and the red dots are gene expression overexpressed genes in tumor cells that are in low TIL areas. And I put a couple of genes here which were interesting. This is zinc protein coding gene, and this is a DNA heat shock C2 gene. Both of them are known to play an important role in activating immune cells. However, I was expecting this to be present here, but not here. This is actually interesting to see. So we need to further validate this and then see whether this has a different function in our system, whether these in a way inhibit immune cells, and that's why we have low tumor infiltrating lymphocyte in this area as compared to this. Now, all this information will be used eventually to compare with a lymph node sections just to make sure if any of the genes are common. If genes are common, then that means those genes are responsible for a lymph node metastasis. So that will be our next step. So overall, all this information will tell us the pathways or genes that are involved in suppressing immune cells and also lymph node metastasis. And I told you in the beginning that these are tumor naive. All these patient samples were collected before treatment was started. Now we are following up with the treatment plan, and these data can be compared with treatment outcomes, and then identify what sort of immune cell profile results in better treatment option, and what sort of a gene expression profile results in better or worse treatment option. Again, contributing towards precision medicine. With that, I'm almost done. So the take-home message is that precision medicine, it's in its honeymoon period now because it's its own challenges, but then it will be a game changer eventually if we address all the challenges. And the information about genetic mutations or protein expression or any other information that we obtain through genomic analysis will be very beneficial in being proactive towards treatment plan. And also it will help us select individual, kind of right treatment plan or personalized treatment plan for one particular group of patients, which will eventually result in better treatment response and also improve their quality of life with limited or less side effects or adverse effect. Now, going back to the challenges that I posed earlier, identification of new gene or driver gene pathways for metastatic breast cancer, I can say that we have some information about that. I mean, you cannot do everything under the sun, but then at least I can say that we are trying to contribute towards, it's a small contribution, but then we are contributing towards development of precision medicine. So far, we know that the signaling pathway, FGF2-DAC1 result, is necessary for metastatic colonization, and targeting them would be beneficial. And now we need to see whether this pathway is activated in all the patients or only a small group of patients. Next, I didn't have time to talk about molecular alteration for specific resistant clones. We have conducted CRISPR Cas9 whole genome screening to look at radio-resistant genes, and then contribution towards identification of biomarkers for diagnosis and prognosis. We have been using 3 different blood-based biomarkers to define oligometastatic state, especially identify oligometastatic patients, so that these patients can be treated with SABR and potentially cured. The last one is, here I got that right. So mechanism of immune suppression that are mainly induced by tumor cells. And to identify different mechanisms, we have performed spatial transcriptomics. Still, it's in a very early stage. We are looking at interaction between tumor cells and immune cells to see what does what and how we can use that information to design better therapeutic strategies to activate immune cells or activate anti-tumor immunity to eventually eradicate tumor cells in the body. With that, I would like to acknowledge my supervisor, Dr. Alison Allen, and my co-supervisor. He's a radiation oncologist who is leading the clinical trial. And I'm also working with breast cancer surgeon on spatial transcriptomics and also CRISPR project. He's also part of clinical trial. And we have collaborators who from Stanford and Amsterdam who are helping us with ctDNA sequencing and immune cell profiling. And this is our lab. And these are some of the other collaborators and some of the funding agencies. And finally, I would like to thank, again, I would like to thank the Institute, organizing committee, technical support, and I would say I was really lucky to have teachers, especially Ramesh sir, who always motivated me and encouraged me to do what I liked the most, and I'm really grateful and really thankful for that. And with that, I would like to end and thank you all for listening. Thank you for your patience, and I apologize for all the technical trouble in between that you experienced. And if you have any questions, I'm happy to answer them. Thank you.
Thank you so much. That was an excellent presentation on the precision medicine against cancer, especially the diagnosis part. As you already mentioned, the loopholes in understanding cancer, pathophysiology, and therapy. We want to take this platform to thank wholeheartedly all the researchers and funding bodies for their interest in research. With your permission, we will take a few quick questions.
Thank you.
You go to sir.
Yeah.
So hold on. You have 3 questions. It's not about the mic. It's working. I have 3 questions to ask. The first question is where you had mentioned about ctDNA sequencing. So just I'm curious that you had done the sequence. Can you just comment that what kind of mutation do you see here? Or is it a particular kind of mutations which had come across some kind
So we are looking, right? We are trying to find. So we are using a panel. We are performing deep sequencing. It's called as cap sequencing, which has pre-made panels, and we know these panels has known mutations, so that are particular to one particular type of tumor. So this study is a multi-histology study. So we have different panels for different types of cancer. I hope I answered your question.
Yes. Second question is regarding the extra vesicle or the exosomal vesicle that you mentioned that they use in it. So if I have missed, can you just repeat that what these extra vesicles do contains, which makes them more prone to go to the lung cancer or the lung tissue or in the case of breast cancer?
That's a really good question. So we, one of the master student is actually working on that. What I can tell you is that it actually, there's a gene called as RABGG2B that in a way, that's what we think that promotes migration of these extracellular vesicles towards lung. We have seen that if you take a cell, breast cancer cells that have higher propensity to go to lung, bone or brain, if you collect EVs from those, they usually do not go to lungs, they only go to bones. So there is a preference. So that means the decision to go to either lung or bone or brain is predetermined when the actual EVs are made. That's what we are thinking. And also it depends on the content of EVs. That's what we think. We are still working on those things. We are trying to look at different properties of EVs to identify. We have done, I think it's done. We have done RNA sequencing on EVs that are produced from different cell lines, breast cancer cell lines. We don't have the data yet, so it's one of the master's project. She's working on that. But there are a few microRNAs that play an important role. I think one is, it's not a microRNA, but RABGG2B is a gene that has something to do with preferential not metastasis preference. It has a preference to go to lung and remodel lung. That's what we think, but we don't have the complete information about that yet. It's still in progress.
Last question. I'm just curious that how would you like to comment that how is this different, this EV than the asides, which you later find on the cancer, later stage of the cancer, some cancer like breast cancer also you find. So how is it different in the EV and the asides?
So EVs and?
So it's basically the fluid filled thing which the later stage of the cancer for breast cancer also we do see the asides or asides.
Asides.
Yeah.
Oh, asides. Well, I don't know the right answer for that. Asides are usually, I've seen that in ovarian cancer, if I'm not wrong, right? Acinized structures, that's what you're saying?
Acinized structure, I was just going through an article that in the breast cancer also, do you find the asides or no? In the latest stage of the cancer?
I don't have that information. I haven't explored that.
Okay.
Yeah.
Thank you.
Thank you. So I have some two questions from the students here. The question is how NARF and HIF1 factors play important role in breast cancer stem cell recognition.
Sorry, could you repeat that?
How NARF and HIF1 play a role in breast cancer stem cell recognition.
HIF1?
Yeah.
So that HIF protein, hypoxia related,
Yes.
right? How they contribute towards breast cancer progression?
Yeah.
Okay, so hypoxia plays an important role. With respect to hypoxia in the primary tumor, I cannot say a lot because my work is mainly focused on metastatic microenvironment. But what I can say is that these proteins, in a way, helps in secretion of different factors such as VEGF, which promotes neoangiogenesis. Then you have TGF-beta, which promotes epithelial to mesenchymal transition, which promotes invasion of tumor cells from its primary site to other parts within the breast. So there's a local invasion and formation of new tumors. And TGF-beta is also known to activate fibroblasts. So when fibroblasts are activated, they again results in proliferation, and these fibroblasts attain activated state and secrete a lot of cytokines, which fuel further progression of primary tumor. Yeah, I guess that's all I can say about that, HIF1 alpha. But then that's mainly related to hypoxia. And also one other thing I can say is that HIF1 does promote excess release of EVs, extracellular vesicles. That's another thing that it does.
And so there's an additional question. Are there any preventive measures to inhibit breast cancer inheritance?
Preventive measures. The only thing is that I think the best example is what's her name? I forgot. Brad Pitt's wife, Angelina Jolie. Yeah. So she had BRCA1 mutation, right? So when that was determined, again, you can say it's an example for precision medicine. She had to undergo mastectomy. She underwent double mastectomy. So that was one of the way how she prevented breast cancer and also ovarian cancer. Yeah.
Thank you. There are any questions? Yes.
Good evening, sir. Sir, ನನ್ನ ಕ್ವೆಶ್ಚನ್ ಬಂದು ಇತ್ತೀಚೆಗೆ ತುಂಬಾ ಕ್ಯಾನ್ಸರ್ಸ್ ಅಂದ್ರೆ ಈಗಿನ ಜನರೇಷನ್ ಅಲ್ಲಿ ತುಂಬಾ ಕಾಮನ್ ಆಗ್ತಿದೆ ಕ್ಯಾನ್ಸರ್ ಸೆಲ್ಸ್ ಎಲ್ಲ ಪ್ರಾಬ್ಲಮ್ಸ್ ಗಳು. ಸೊ ಇದ್ರ ಮೇಲೆ ತುಂಬಾ ರಿಸರ್ಚ್ ಗಳು ಕೂಡ ನಡೀತಿದೆ. ಬಟ್ ಪರ್ಮನೆಂಟ್ ಆಗಿ ಕ್ಯಾನ್ಸರ್ ಗೆ ಮುಂದೆ ಇನ್ ಫ್ಯೂಚರ್ ಅಲ್ಲಿ ಯಾವ್ದಾದ್ರು ಟ್ರೀಟ್ಮೆಂಟ್ ಬರೋ ಸಾಧ್ಯತೆಗಳು ಇದೆಯಾ ಸರ್? ಯುವರ್ ಒಪಿನಿಯನ್.
ನನ್ನ ಒಪಿನಿಯನ್ ಪ್ರಕಾರ ಇಮ್ಯುನೊಥೆರಪಿ ದಟ್ಸ್ ಅ ಗೇಮ್ ಚೇಂಜರ್. ಸೊ ಈಗ ನೋಡಬೇಕಾದ್ರೆ ಇಮ್ಯುನೊಥೆರಪಿಯಲ್ಲಿ ತುಂಬ ಡೆವಲಪ್ಮೆಂಟ್ ಆಗ್ತಿದೆ. ಇಮ್ಯೂನ್ ಸಿಸ್ಟಮ್ ಇಸ್ ಇಂಪಾರ್ಟೆಂಟ್ ಅಂತ ಹೇಳಿದ್ನಲ್ಲ. ಸೊ ಇಫ್ ಯು ಫೈಂಡ್ ಅ ವೇ ಟು ಆಕ್ಟಿವೇಟ್ ಇಮ್ಯೂನ್ ಸಿಸ್ಟಮ್ ದೆನ್ ಇಟ್ಸ್ ಗುಡ್, ಬಿಕಾಸ್ ಕೀಮೊಥೆರಪಿ, ನೀವು ರೇಡಿಯೋಥೆರಪಿ ಆದರೆ ಅದು ಎಕ್ಸ್ಟರ್ನಲ್ ಸೋರ್ಸ್. ಸೊ ಇಮ್ಯೂನ್ ಆಕ್ಟಿವೇಶನ್ ಅಂತ ಹೇಳಿದ್ರೆ ನಿಮ್ದು ಅದು ಹೋಸ್ಟ್ ಇಮ್ಯೂನ್ ಸಿಸ್ಟಮ್ ಅಲ್ವಾ. So it's same as activating immune system against any other pathogens. So I believe immunotherapy would be the next treatment modality to treat and cure cancer. But one disadvantage ಏನಂತ ಹೇಳಿದ್ರೆ, it will not replace the traditional classical therapy options like surgery or radiotherapy or chemotherapy. So immunotherapy should be given in combination with those things. That's one thing. One drawback with immunotherapy ಅಂತ ಹೇಳಿದ್ರೆ, the patient needs to be monitored. If they are on immunotherapy, 24-hour observation ಇರಬೇಕಾಗುತ್ತೆ. ಯಾಕೆ ಅಂತ ಹೇಳಿದ್ರೆ, ಯಾವುದೇ ಕಾರಣಕ್ಕೂ ಇಮ್ಯೂನ್ ಸಿಸ್ಟಮ್ ಓವರ್ ಆಕ್ಟಿವೇಟ್ ಆದರೆ it will damage all your organs. So some of the patients who are under immunotherapy, they have adverse effects. So adrinda I would say it is the next thing, and it might cure cancer, but still it will not be used as a frontline therapy option, or it might be used as a frontline therapy option. But in another 25 years, I see that immunotherapy will be the next therapy option to cure cancer.
Thank you, sir.
So it's only by query.
Hello, sir.
Right. Immunotherapy leads to several kinds of side effects. Basically, when monoclonals are used for therapy. Can you hear me?
Yeah, I can hear you.
Yeah. Apart from that, now the trend is to generate CAR T cells and then use it for various kinds of cancer therapy, and it's a promising one. What do you think whether this CAR T is being tried in Canada already? Because there are many reports CAR T cell therapy has quite a bit alleviated cancerous conditions. In Germany and also in the US, that is one of the latest technologies that is being used. Whether it is in practice in Canada.
For blood-related tumors, hematological tumors, it's easier to use CAR T cell therapy. But for solid tumors, it's a bit challenging, because the CAR T cell has to in a way infiltrate the tumor, and that's one of the major challenges with respect to solid tumors. And with respect to there are some clinical trials that are in progress. One of the clinical trial in our institute, I don't think they are doing PD-L1 therapy on lung cancer using CTC as a biomarker. But others in our institute, I don't see. But then in Toronto, I believe they are trying out something. But again, it's in clinical trial stage. The best example for CAR T cell therapy is Emily Whitehead, and she's still alive. It's been 10 years.
More than that, it's right now it's very expensive.
Yeah.
$450,000 per treatment.
Yeah.
So that is the limitation also.
Yeah, that's also another limitation. Yes, definitely.
Yeah, but now successful results have been obtained with regard to autoimmune diseases, prevention of autoimmune diseases, like these CAR T cells.
Yeah.
Different kinds of approaches or a combination of many approaches.
Yeah. And also they are looking at gene editing technology to cure, some of the diseases.
Yeah.
Yeah.
So before I hand over the mic, I would like to profusely thank you personally for
Thank
being
you.
here with the late night for you.
Oh, that's fine.
If there are some more questions from the students.
Good morning, sir.
Hello.
My question is on CRISPR-Cas9. So, if I'm not wrong, suppose if it is a metastatic cancer, so if there's a breast cancer, so the cancer which is caused by the gene, that is BRCA gene and all, the mutations. So, if it is caused by mutations, is it possible, like in CRISPR-Cas9, if I'm not wrong, so we can create our gene of interest and we can insert into the genome and we can cure the diseases. So is it possible, like if it is a metastatic cancer, if I take breast cancer only, so editing that mutated gene, so if it is effective that we can cure that disease permanently?
Well, hypothetically, based on recent results, I can say that gene editing might be useful, might correct mutation in those things. But then in the long run, I don't know what's going to happen, whether that leads to another complication. We don't know yet. So hypothetically, it is possible.
I just curious about is it happened while conducting experiments on mice models?
With respect to cancer, I don't have information, but for other diseases, recently they did it. So that was successful. So based on that information, that's the reason I said it is possible. We have the technology to do that. But then I don't know how patients respond. Because you're manipulating a system, so you need to be extra cautious.
Thank you, sir.
Yeah.
Thanks a lot for joining us late night. I think it's early morning now. Thank you so much. Thank you so much.
Thank you. Thank you.
Thank you so much, sir.
Thank you. Thank you for having me. I hope it was useful. Thank you.